Merci Claire. Si ça dérange personne, on va faire en anglais parce que je pense que ça va être mieux pour tout le monde. So, I'm going to talk about viral protein expression and the modulation of DNA repair, and much more specifically about liver cancer, hepatitis or HBV, and radiotherapy. Okay, let's try. Oh, how do we make this move? Oh, I've killed it. That one? Okay, so declaration of interest, Anexo pays some of our studies. So, hepatocellular carcinoma, it's the 5th most common cancer for men and the 7th most common in women. And as you can see from this map, there's a very uneven distribution in mortality around the world. The risk factors for hepatocellular carcinoma are hepatitis B and C virus, alcohol, aflatoxin B1, and metabolic disorders. This is not working. Oh, this one. Okay, got it. So, for hepatocellular carcinoma, there's very few treatment options. Radiotherapy is becoming a very promising approach. In particular, there's been some studies ongoing in France looking at serotoxic irradiation. And as you can see on the slide, the curative rate for small cancers is actually quite good, about 95%. But for the larger tumours, the response is much poorer. And so one of the questions we raise, as Mary has been explaining to you, is whether the radioresistance could be due to an elevated DNA damage response in the liver cells. So to look at this, we wanted to see whether radiation sensitivity could be increased, as Marie explained to you using her debate, by inhibiting DNA repair. And so we wanted to try to investigate whether DNA repair inhibitors were really an option for HCC treatment. And as Marie, we were particularly interested in looking at in two... Oh, gosh. Where's the pointer? So we were particularly interested... yes, it does work. We were particularly interested in the DNA repair enzymes that are involved in single-strand break repair and double-strand break repairs, because, as you've already heard, these are the two lesions that are probably the most toxic generated by radiation therapy. So we were particularly interested in part 1 and part 2, DNA PK, and ATM. So as many other people, when you start to do these type of investigations, you turn to what's available in the databases to get some first idea of what's out there in terms of information about liver cancer. And we looked at the TCGA provisional database, which is available via the CBO portal. And I made my list of favorite DNA repair enzymes, and I looked to see what was different, whether these repair enzymes were expressed differently in these liver tumors. As you can see, there's quite a bit of variation. The red bars are messenger RNA activation overexpression. What really struck me was that the PARP1 gene, messenger RNA, was overexpressed in about 33% of tumours and DNA PK in 23%. Now, as you've heard already, these two particular DNA repair enzymes are both druggable and, when you inactivate them, cause radiation sensitivity. So we also wanted to know a little bit more about what was happening, because those samples there, you can't actually get the viral information. In the databases, it's actually very difficult to be able to tease out whether the liver cancers are due to HBV, HCV, or alcohol. Luckily, in the lab, we have access to, via the National HCC Biobank, a panel of tumours which were of different etiology, 20 liver tumours which were HCV related, 21 HBV, and 19 alcohol. And RNA was isolated from all of these tumours, and we used, in collaboration with Yvon Biersch and Sophie Vacher Institute Curie, we analysed my panel of 17 favourite DNA repair enzymes to look to see whether there were really any differences in expression in this panel of tumours. This is the heat map of the expression profile, and you can see red is overexpressed. So here we have the HCV tumours, HBV, and the alcohol-related tumours. And down here we have our control panel. And the heat map is relative to 3 times the standard deviation levels of expression we're seeing in the controls. And what's really very obvious is that in all of these tumours, there's overexpression of many of the different DNA repair enzymes. As I said, we were particularly interested in PARP1 and DNA-PK because these are two where there are very good small molecule inhibitors that are available. And so here is the data for PARP1, here for DNA-PK. This is the normal liver, and here we have the tumour and the peritumoral tissue for those tumours in pairs, HBV tumours, the HBV normal tissue surrounding the tumour tissue, the alcohol, and the alcohol normal tissue. What's intriguing to me is that it seems to be it's within the tumour tissue, not the peritumoral tissue, that we're getting these increases in PARP expression, and quite clearly the highest increases are being found in the HBV-associated liver tumours. DNA-PK has a similar sort of profile, with again HBV-associated liver tumours having the highest level of expression of DNA-PK. So what are these PARPs? The PARPs are a family of 17 enzymes which are catalyze ADP rearboxylation. And they were identified because they all have homology to the catalytic domain. So they were identified, some of them by bioinformatic approaches, and as I say, all share the same NAD substrate recognition. PARPs1, PARPs2, and PARPs3 are all involved in mammalian cells in the repair of strand breaks. PARP1 and PARP2 more single strand breaks, PARP2 and PARP3 are both involved in the repair of double strand breaks. So the inactivation of these proteins by an inhibitor would lead to radiosensitivity. So the PARPs, what they actually do is they bind to the, when we generate a single strand break in the cell, PARP1 binds, its activity is activated. It uses NAD to generate these sort of little molecular webs that hang around onto the PARP1 and many other proteins. And these are used to attract the other DNA repair proteins to the site of the damage. These polymers are negatively charged. The DNA is negatively charged. And so the production of the polymers on PARP itself results in the PARP1 coming off the DNA and leaving the other DNA repair enzymes to do their job and repair the damage. PARP1, the polymers on PARP1 are now degraded by a protein called PARG, and then PARP1 can be recycled. So this is a structure of function of the structure of PARP1. So PARP1, as I said, binds first to the strand break using its zinc fingers. This binding to the strand break reduces a conformational change and opens up the active site and allows the binding of NAD. This bond here in the NAD is cleaved, liberating nicotinamide, and this is the polymer which is then going to be attached to the proteins. The first inhibitor of PARP which was actually used was 3-aminobenzamide, and as you can see from the structure here, it very much resembles nicotinamide, and it binds very efficiently into the active site. But this inhibitor, which will inhibit PARP1, PARP2, and PARP3, and in fact all the PARPs, because they've all got the same active site, wasn't specific or potent enough for more advanced clinical trials, and we had to wait for second and third generation catalytic inhibitors to be produced, which I'll show you in a minute. But just as a side story, if you want to work with PARP inhibitors, don't use this one. This is the one that actually went into phase 3 clinical trials, but unfortunately doesn't actually block the catalytic activity of PARP. Iniparib binds to the zinc fingers, and so what's going to happen here is that it's going to prevent PARP binding to DNA. Why is that important? Well, if you don't bind it to the DNA, these inhibitors will actually end up blocking PARP onto the DNA. If we go back 2 slides, PARP activity is generated once it's been bound. So if you don't bind, you can't generate the polymers. If you bind and then inhibit, the PARP1 will rest blocked onto the DNA. And that blockage is actually really important for the cytotoxic effects we're going to see. So these are the PARP inhibitors that are presently in clinical trials in a variety of phase 1, phase 2, phase 3, but really nothing very much happening in liver cancer, and not so much combined with radiation. So what's the mechanistic basis of PARP catalytic inhibitors combined with radiotherapy? PARP inhibition, as I've shown you, will block the repair of single strand breaks. This will generate more double strand breaks when you've got replication. We're going to trap PARP1 onto the DNA because we're going to allow it to bind to the DNA. We're going to block its activity. it can't come back off the DNA very easily, and it has to be actively repaired to come off, and some of those repair mechanisms require homologous recombination and other repair enzymes. There's also a very important factor of these PARP inhibitors, so they will also alter the tumour vascularization. They will improve blood flow and reduce hypoxia within tumours, and if you have a reduction in hypoxia, you will get an increase in radiosensitivity. So it's a combined effect, not just on DNA repair, but we believe there's also quite an important component on this tumour vascularization, which is going to be important. Now, if we do add, as well as the PARP inhibitor radiation, we're going to generate even more damage, which is going to be even more difficult to repair. So the proof of concept studies were done in the lab when I was still in Paris in collaboration with Isabelle Chemin and Clément Guillot, who was doing a PhD in the lab at the time. And we showed here that this is just treating a panel of liver cancer cell lines with 2 hours treatment with PARP inhibitor as a single agent. We use ABBA-888, which is also known as veliparib. It's a catalytic inhibitor. And you can see that the clonogenic survival was reduced, not very markedly, but in sometimes a little significantly, after just a 2-hour treatment with the inhibitor. So we then took 2 of these cell lines, the HepG2 and the PCL5 PCRF5 cell lines, and we treated those with inhibitor and radiation. So if we look at the top 2 lines here, this is the cell survival of HepG2, and HepG2, where we've given the cells 30 minutes before the irradiation dose a PARP inhibitor. And you can see that there is a reduction in their cell survival in the presence of the PARP inhibitor at the time of radiation. This is the second cell line we looked at, and you can see already the cell line is much more radiation sensitive than the HepG2, and there's also a very marked decrease in the radiation sensitivity. We were a little intrigued by this, but didn't really think too much about it at the time until we went on to look at several more panels of cell lines. And here, what we've done here is, again, using clonogenic survival, treated the cells either 2 hours or 24 hours of PARP inhibitor, and looked at the survival. Some cell lines, there's really no effect. HEP3B, for instance, we don't see any effect of the PARP inhibitor over a 24-hour exposure. This cell line has been defined as a very homologous recombination proficient cell line, and I'm wondering whether the PARP inhibition, the PARP inhibitor that's being blocked onto the DNA is actually being very efficiently repaired, so we can't see an effect of the inhibitor on its own. What was very intriguing is when we compared the HepG2 to HepG225s. This cell line here is a cell line that's expressing viral proteins, and it's actually the same genetic background as this one. So what we can see is in the cell line that's expressing the virus, in the same genetic background, the effect of the PARP inhibitor is much more marked. This intrigued us a bit, so we started looking into this into a bit more detail. So this is comparing the effect of the ABBA-888 with ionizing radiation, the HepG2 cells, plus or minus radiation. If we have the HepG2 cells with the inhibitor present with 2 grays of radiation, we get a 6% gain in survival in the presence of the inhibitor. With HEP3B, it's larger. HEP3B cells also have viruses. So we now had 3 cell lines which were showing a larger effect when we're having virus being expressed. Obviously, we've got lots of differences in genetic background, so we couldn't conclude anything from this. So we then resorted to another pair of cell lines in the lab, which is, again, the HepG2-225, and this is a cell line carrying the HPV wild-type genome, and then a second cell line, which is called HepG2K6. And this is a cell line which carries a deletion within the HPV genome, which expresses no HBx protein. So what we can see, if we look here, these are the cells which are just treated with the PARP inhibitor, 2 hours and 24 hours. In both cell lines, we see a decrease in survival, but in every time when we have the wild-type genome being present, we have a larger decrease in expression, in effect on the cell killing. The same response of ionizing radiation. We have a difference between, and each time the difference is larger when we have the wild-type genome. So this was leading us to suggest that there is an effect, and the effect could be somehow related to the protein, HBx protein, which is increasing, which is present in the cell line. So our conclusion from the experiments I've shown you is that the expression of the HBV-encoded HBx protein increases the sensitivity of liver cells to the cell-killing effects of both ionizing radiation and PARP and the combination. So what's the mechanistic basis? And this is where I was really pleased to come to the lab, because they've been working on this for years. And this is the result of a beautiful collaboration that's been done by the people in the lab, Laetitia, who's here, and Olivier, who's here, and they can answer all the virus questions, not me. And they've done this collaboration which shows that the HBX virus protein identifies the SMC5 and 6 complex as a host restriction factor, which all sounds very complicated, and I'm going to try to explain. So, we have the HB virus that's present and can infect hepatocytes and be present in the nucleus. Its DNA is present in the nucleus as a form of cccDNA. And what the guys in the lab showed, together with their collaborators from Geneva and Gilead, was that the SMC5-6 complex is present on this cccDNA and blocks the transcription of the cccDNA. To be able to have viral replication, they have to remove the virus, wants this to be removed from the cccDNA to allow this to become active to produce more virus. And it's developed a really clever way of doing it. It hijacks the SMC5 and 6 proteins from the cell. So what happens is, using the HBX protein. So the HBX protein binds to DDB1, and here I have to put my glasses on, binds to DB1, and this complex binds to the SMC5-6 complex and takes the SMC5-6 complex towards the ubiquitin ligase. This will degrade SMC5 and 6, removing it from the cccDNA, and the cccDNA is now active. Now, this is perfect for the virus. The virus is now being able to replicate, but what's the consequences on the host cell? The host cell has now lost its SM5 and 6. So what are these SMC5s and 6? Eukaryotic cells have 6 SMC proteins. SMC1 and 3 form the complex called cohesin, which is absolutely required for chromosome segregation. SMC2 and 4 are condensin, which mediates chromosome condensation during mitosis. And SMC5 and 6 has multiple roles in DNA repair. And if you start looking in the literature, it gets about as vague as that. Multiple roles in DNA repair. It's not really well defined what the phenotype of these cells are. There are many papers that suggest it's involved in double-strand break repair using sister chromatid recombination. It's involved in replication fork rescue, and it's involved in RNA, ribosomal DNA integrity. And if you start looking at all these structures that you're finding here, some of them will resemble the structures that we will need to resolve if we're actually repairing a blocked PARP protein or a damaged a block part protein which has been trapped onto DNA. So our working hypothesis at the moment is a loss of the SMC5-6 complex, because the fact the virus is expressing the HBX protein, enhances the cell killing effects of the PARP inhibition and ionizing radiation. And this is the graph I showed you before, and here we have the Western blot that corresponds. So this is the 225 cell line, which is carrying a wild-type genome and has no SMC6. When you destabilize SMC6, you destabilize the rest of the complex. Whereas the K6 cell line, which is carrying, which is deplete, has the X being present. So what we were interested in doing now was whether the expression of viral proteins will also sensitize to other drugs. Obviously, we've got an overexpression of DNA PK, and there are DNA PK inhibitors out there, and we're going to plan to check those. Obviously, it's going to be very interesting to look at debate and see what happens there as well. And the other thing we're interested in looking is looking at HDAC inhibitors. HDACs are absolutely essential. They balance the acetylation, deacetylation profile, which is absolutely essential for the chromatin environment in cells. And they facilitate DNA damage response by loosening the chromatin. HDAC inhibitors are going to block the chromatin changes which are needed for repair and inhibit certain repair enzymes. So we've just started looking at the effect of HDAC inhibitors, and this is the first slide with a set of results. What we did is we plated the cells, we treated them for 24 hours with the inhibitor, gave them ionizing radiation, removed the drug 24 hours, at which point we can see that we've got an increase in histone H3 acetylation. We have an increase in the DNA acetylation. And these are some of the flasks from the cell survival. So we can see that if we give the inhibitor on the own, we seem to be having a major cytostatic effect more than perhaps cell-killing effect. But when we combine the inhibitor with the radiation, we're getting a very nice effect on cell killing on these cell lines. Obviously, we need to go back now and do all those experiments with the 2 lines expressing the HBX and not HBX to see whether we're seeing the same effect on these. So, in conclusion, I hope I've managed to convince you that using classical PARP inhibitors might actually be very useful as an adjuvant to radiotherapy in HCC, which clearly are expressing PARP1. HCC cell lines in our hands seem to be sensitive to the combined effects of PARP inhibitors and radiotherapy, and cell lines expressing these viral proteins clearly show an increased sensitivity to PARP inhibitors and HDAC inhibitors. And we believe that part of this is due to the role of the HBX protein and its impact on the SM5 and 6 complex in the DNA repair pathway. And these are the guys that have done all the work, the people in the team here in Lyon, which is led by Isabelle Chemin and Philippe Merle, Yvon Bièche and Sophie, who've been helping us with the messenger RNA, the radiotherapy department at Centre Lyon-Bérard, who allow us to come and irradiate ourselves with them and not ask too many questions about what we're doing, and we try to explain. Michel Strubin, his colleagues, and we'd like to thank the League for their financial support. Thank you.